Welcome to the Discovery Dialogues podcast, where we take a deep dive into the history of groundbreaking discoveries in the life sciences. I'm your host, Ani Deshpande. Each episode, we will uncover some incredible, insightful, and often just insane stories behind breakthroughs in medicine. Join us as we journey through the twists and turns of scientific history, one breakthrough at a time. In our first episode, we are diving deep into the history of one of the world's oldest known diseases, diabetes. Join us as we explore how ancient Egyptian texts provided some of the earliest descriptions of this condition and why physicians in India relied on the trails of ants. We will also discuss how a curious Lithuanian immigrant collected the urine of an incontinent dog to make an astonishing discovery. We'll also see a German scientist who used industrial dyes to color organs. And finally, we will uncover the story of an eccentric Canadian whose work led to the biggest blockbuster drug of all time. From ancient manuscripts to life saving breakthroughs, we will unravel the fascinating journeys of how we came to understand diabetes. To travel with us through the history of diabetes and understand the new exciting developments that have taken place, I'm pleased to have my colleague, Dr. Pamela Itkin Ansari. She has done research on diabetes for about 2 decades, research that has gone on to clinical trials. And Dr. Ansari has also been a board member of the Juvenile Diabetes Foundation and has given a TEDx talk. I'm really jealous of you, Pam. Why don't you tell our audience what diabetes is?
Okay, thank you, Ani. I'm very happy to be here. I'm a great admirer of your research and your scientific prowess. So thank you for inviting me. Diabetes, whether type 1 or type 2 or gestational diabetes, it's all a disease of how the body utilizes sugar. I think let's start this episode by learning how diabetes was identified as a disease by its symptoms.
All right, so let's dive in. And diabetes is by no means a new disease. In fact, it has a long history spanning across multiple ancient cultures, including Egyptian, Indian, Chinese, and Middle Eastern civilizations. What's fascinating is how these cultures systematically observed patients, grouped them on the basis of common symptoms,
Right.
and eventually identified what we now come to recognize as diabetes. In fact, one of the earliest records of this disease comes from the ancient Egyptian documents called the Ebers Papyrus. This text, which dates back to, believe it or not, 1500 BC, was acquired by the German Egyptologist George Ebers, hence its name. And when scholars translated the script, they found descriptions of patients exhibiting symptoms remarkably similar to what we now diagnose as diabetes. And the Ebers Papyrus refers to wasting disease with the propensity to, quote, eliminate urine, which is too plentiful. So fast forward to roughly 600 BC, more than 2000 years ago, the ancient Indian texts have a very detailed record of this condition, which the physician Sushrut is often regarded as the father of surgery. In fact, I have a book here, the English translation of the Sushrut Samhita, and this book is more than 2000 years old. This not just lays the foundation for plastic surgery and has things such as nose reconstruction or rhinoplasty, but also documents many diseases, including, as we will see, diabetes. In fact, the Sushrut Samhita describes patients with an unquenchable thirst, what we now call polydipsia, and frequent urination or polyuria. Another truly remarkable part is that the Sushrut Samhita and later the Charak Samhita, which is another later ancient Indian text, and these texts form the crux of what we now call the Indian medicine system of the Ayurveda. They noticed that the urine of these patients have a sweet quality, and we, of course, now call it glycosuria, the presence of sugar in the urine. In the 2nd century BC, the Huangdi Neijing, or the Yellow Emperor's Inner Canon, a foundational text of traditional Chinese medicine, has detailed descriptions of a wasting disease with excessive thirst, frequent urination, and an unexplained weight loss.
So what you're telling us is that at least 2,000 or 3,000 years before modern medicine, ancient physicians were already identifying the symptoms of diabetes. It's incredible to see how early medical minds making observations that would eventually help shape our understanding of the disease.
Well, there was another surprising hint. In ancient and medieval doctors would actually taste their patient's urine to diagnose the disease. It sounds shocking today, but back then it was a legitimate diagnostic method. And they found that the urine tasted sweet. And these ancient physicians also observed that the urine, when left out to dry, when exposed to the elements, would turn sticky. And the Sushruta Samhita also has a very surprising observation that the urine of patients with this condition would attract insects and particularly ants. And this reinforced the idea that there was something sugary present in the urine.
Right.
This was an astonishing insight for its time. And so the Sanskrit name for this disease is actually Madhumeha. Madhu, which means honey or sweet, and meha, which means urine. So the name in Sanskrit actually means honey urine.
Right.
Pam, can you help us understand why would diabetic patients feel so thirsty and have the urge to frequently relieve themselves? And what does this disease have to do with sugar in the first place?
Right. So first, sugar is the major fuel for the cells in the body, and it's the end product of much of what we eat. Without sugar, the trillion cells in our body don't have the energy they need to grow or carry out normal functions. What these people were observing was the result of first, the cells not being able to take in the sugar they needed. And so it was second, spilling out into the urine. When blood sugar runs too high, the kidneys clear out the excess in urine, but sugar pulls with it liquid. And so this led to the frequent urination and dehydration. That's why the patients with diabetes felt constantly thirsty. Their body was trying to replace the lost fluids. It's often the first red flag that something's wrong with blood sugar regulation, even now. So if we think of a car analogy where an empty gas tank needs refueling, then diabetes is like trying to pour gasoline into the engine, but the fuel line is blocked. The gasoline would just spill onto the ground. And of course, the car won't be able to move.
It's incredible to think how early physicians, using just simple but really elegant observations,
Right.
were already laying the groundwork for understanding diabetes long before modern biochemistry confirmed their insights. And if in Sanskrit it's called Madhumeha or honey urine, and it had different names in different cultures, how did the name diabetes mellitus come along?
Right.
So Apollonius, the Greek physician who lived in Egypt in Memphis around the 2nd or 3rd century BC, coined the term diabetes. This term means to siphon or to pass through, and he was, of course, referring to the intense urination that characterized this condition, as if the body was just constantly draining itself of fluids. The disease also finds prominent mention in the Greco-Roman physician Artius, who was in the 2nd century AD, and in many Chinese and Arabic texts. All these influential medical thinkers also wrote extensively about patients with excessive thirst, frequent urination, and the unexplained weight loss that accompanies this condition. Now, one towering figure that we need to mention is Ibn Sina, who is this Persian polymath, also called Avicenna by the Europeans. He worked under various Persian and Arabic rulers, and he had a deep fascination with medicine. He's widely considered one of the most influential philosophers and thinkers of his time in the Islamic world. And he compiled knowledge from Greek, Indian, Chinese, and Middle Eastern medical traditions into this one very influential book called the Al Qanun fi al Tib, or it's called in the West the Canon of Medicine.
Right.
What is remarkable is that Avicenna didn't just document diabetes. He went a step further and he recognized the complications of this disease, such as nerve damage and gangrene, which was really amazing for that time. Now, the term diabetes remained in medical use until centuries later. The full name diabetes mellitus was introduced. The Latin word mellitus, meaning honey sweet, was added later in the 17th century by Thomas Willis, who was an English physician. The current word for the disease, diabetes mellitus, which is derived from Greek and Latin, is actually almost exactly the same as the Sanskrit word madhumeha.
So it didn't really change over time. And Ani, one of the most fascinating aspects of the history is that it wasn't isolated. physicians and scholars across different civilizations did learn from each other, passing knowledge of previous work and that the new discoveries were built upon. Even though they lived centuries apart, medical thinkers were part of a long chain of intellectual exchange, each refining and expanding on the observations of those before them, just as we do today. However, there's a key difference because then it was painfully slow. It could take years or centuries because knowledge back then traveled along trade routes, often taking so long to reach new regions because it was following the Silk Road or the Arabian Sea trading networks or Mediterranean routes. And even going through merchants and taking painstaking care by certain people to translate and transcribe these observations so that they could be eventually shared globally.
And it would take a few thousand years. That's so different from today, where we have instant access to information and global collaboration, and we can share and build upon discoveries almost instantly.
Exactly.
And I also want to make an important point here that at various times in history, some of the most important discoveries were made in pockets of civilization, such as China and India and Egypt and the Middle East and Greece, where scholarship and scientific inquiry and the exchange of ideas were encouraged. With the Renaissance, these ideals gained prominence in the western parts of Europe and later shaped the broader Western world.
I'm amazed to know that so much was already known about diabetes just from the symptoms. But once you know there's a problem with the way the body processes sugar, how do you find the source of the problem in the body?
Of course, once you recognize the disease by its symptoms, the next big question is where exactly is it happening in the body? And that's where things get really interesting because for centuries, if you're studying a disease, you could only look at the symptoms and you could rely just on the external observations. There were no X-rays, there were no MRIs, and there were no CT scans, of course. So physicians could see the symptoms, but they had no way of looking inside the body. Sometimes they could do post-mortem examinations, but that was about it. So what is the organ that is involved in diabetes? Right. To get to this, I'm going to recount two crazy but true stories. For the first story, you have to remember that to get at any disease, you have to have a knowledge of the human body and its various organs, which is called anatomy.
Right.
And Europe of the 18th and 19th centuries was the leading center of anatomy studies. Physicians in the medical schools were desperately trying to find bodies to dissect. And the problem was that human cadavers were not easy to come by because the dissection of human bodies was considered immoral, unethical, and of course, often strictly forbidden by religious diktats.
Right.
Some laws, for example, require that corpses used for medical research only come from suicide victims
Right.
or from people who had died in prison. And of course, when there is a huge demand and supply gap, what comes in to fill in the gap is a black market.
Not good.
In the early 1800s, professional grave robbers called resurrectionists were robbing graves and selling cadavers to anatomy professors. The study of anatomy is unfortunately rife with many such accounts. And throughout the 18th and 19th centuries, it was really the Wild West in anatomy research.
Right.
This came to a head really in one of the most infamous stories in medical history, where a surprisingly large number of bodies in Edinburgh were being provided by two individuals, William Burke and William
And
Hare.
then
And then it turned out that, well, they were actually murdering their victims and supplying their corpses. And this case shocked everyone, including the scientific community. Their crimes really exposed the reality of the demand for cadavers and led to an important milestone in medical research ethics, the Anatomy Act of 1832, thankfully, which finally legalized and regulated body donation for medical research.
So allowed for the research, but made sure everything was done ethically. Wow. These dark stories remind us of just how far medicine has come and how essential anatomy was and still is for understanding human disease. Thank goodness today, with ethical donations, and more advanced imaging, no one would resort to body snatching. I can't wait to hear the other story.
Well, ultimately, anatomy studies made a very nice distinction between the different organs of the body, but they did not really tell us how these organs actually interact with each other
Right.
in a living organism, a study that then came to be called physiology. And this is a very important inflection point in the history of medicine. That's where I will recount the second fascinating story. And the hero of this story is Claude Bernard, a French scientist from the early 1800s. Bernard is often called the father of physiology because he realized that if you wanted to understand the links between anatomy or the organs and physiology, how these organs talk to each other, you could not do that in humans. You needed to do experimentation in animals.
Exactly.
And so he really underscored the importance of animal experimentation.
And I find it really striking that if you were doing scientific research back then, there were no paid scientists who were biologists. Nobody would support that. So you had to find a different way to support yourself.
I know, and in fact, Claude Bernard was not very well to do, but he got married, and his wife was from a very influential Parisian family, and he got a hefty dowry in his marriage.
So that's how he did it. Okay.
Yes, so there are many accounts which say how that dowry actually helped Bernard carry out all the extensive experiments and supported his research. So it can be said that he found a way to fund his science. Now, the really interesting thing here is that his wife, Marie, was really fond of dogs. And when they met, she was delighted to know that Claude was also really interested in dogs.
Right.
And so I think it started really well, their marriage. But soon she realized that his interest in dogs was slightly different from hers. Claude wanted to know, for example, if you feed sugar to a dog and kill the dog immediately, what happens to the sugar? Where does it end up? So Claude Bernard was the quintessential experimentalist.
Okay. What we haven't discussed yet is that whether we're eating or we're fasting, the body needs a set and controlled amount of sugar in the bloodstream at all times. In particular, the brain needs stable levels of blood sugar. So how does the body regulate that?
Well, when Bernard started experimenting with his dogs, he realized that whether the dogs had eaten anything or not, the sugar in their blood and therefore the sugar supplied to the organs would remain somewhat stable. And this perplexed him. This was a mystery.
Yes,
If you don't eat sugar, then where is the sugar coming from?
exactly.
And so, what happens when you are fasting? He realized that when the dogs were not given sugary meals, the sugar supply in the blood seemed to come from a surprising source, the liver. And these experiments suggested that the body was storing sugar in this organ. And the important realization from Bernard's studies was that the organs were not just individual structures, but also worked together in a fine tuned system, constantly adjusting to maintain the balance. This was really a radical idea at
Right,
that time,
right.
but it led to one of his most famous contributions, the concept of what we now call homeostasis.
Exactly.
Now we know that the body has a way of actively regulating its temperature, the level of constituents of the blood, and importantly of sugar and many different things, regardless of the external environment. And this concept of homeostasis was largely due to Bernard's work. Importantly, his work clearly pointed to the possibility that diabetes was not just about excessive sugar appearing in the blood, it was also a dysfunction of how the body regulates and uses sugar. So, amazingly, through these experiments, Bernard was one of the first to notice that not only the gut, but also the brain may play a role in blood sugar control. And so he would do all these experiments on animals and often at home and on his pets. And of course, his wife was aghast. In fact, Claude and Marie Bernard eventually separated, and this was really uncommon,
It
as you know,
was.
in 19th century France, and just shows you how big of an intellectual divide there was between them. And Claude Bernard eventually became a strong proponent of animal experimentation. He really believed that this was important to understanding human disease. And Marie, on the other hand, she spent the later part of her life as an activist who contributed to the early animal rights movement in France, challenging prevailing scientific practices and advocating for more humane treatment of animals.
Both of which I think were very important.
Yes, both views. And the story of Claude and Marie Bernard is actually interesting and important because this also vividly portrays the two contrasting thoughts that continue to this day. One which believes that animal experimentation is so crucial for us to understand and cure human disease
Yeah, done humanely.
as long as it's done humanely. And the other school of thought which believes that any animal experimentation is just immoral and unethical and unacceptable.
So even though Claude Bernard has been celebrated, and he was the first ever scientist to receive a state funeral in France, his contributions were even bigger than was noted at the time. And as we'll see in incredibly exciting episodes that follow, the links between the liver, the gut, and the brain to diabetes more than 100 years after his death are still being understood and studied, and they all harken back to Bernard's foundational work in modern physiology.
I agree. And before we move on from Bernard, it is worth noting that even though he really set out to find the organ that was central to the origin of diabetes, for a while he believed that it might have been the lung.
Yeah.
And he realized later that he was wrong. And even though he had a strong interest in the pancreas, and in fact, his elegant work showed that it was involved in the digestion of food, somehow he missed the connection of diabetes to the pancreas. But there were others who were starting to wonder if there is a connection, whether this little comma shaped organ that is adjacent immediately to the gut plays a role in diabetes. And one of the reasons for this is that there were medical examiners who would see repeatedly in the 19th century that there was pancreatic damage in some diabetics when they did autopsies on these patients.
So how do you prove directly that the pancreas is the organ that's responsible? Well, you'd want to remove it. But at that time, it was very difficult to perform a complex surgical procedure without immediately killing the animal. So this is where Joseph von Mering, a German physician, challenged his surgical colleague, Oskar Minkowski, a Lithuanian immigrant, to try and remove the pancreas. And Minkowski was known for his deft surgical prowess and took up the challenge. And in fact, together, the very next day, they performed the first ever pancreatectomy, meaning removal of the pancreas.
So what happened next was really amazing. Over the next few days, the dog would start urinating uncontrollably and excessively all over the floor. And Minkowski then collected a urine sample from the floor. And upon testing, he found that it was loaded with sugar. He immediately recognized that removing the pancreas had induced diabetes in the dog. This was a groundbreaking moment because for the first time in history, diabetes was now directly linked to the pancreas.
Exactly. Well, I think there's something else that's really interesting in this story. So Minkowski didn't just stop at proving that the pancreas was necessary to control blood sugar, which would have been plenty of a contribution.
Right.
He went a step further to ask, is there any way you could prove that the pancreas was also sufficient? And so remarkably, after removing the pancreas from one dog, he transplanted a pancreas from another dog just under the sick animal's skin. And amazingly, this restored blood sugar back to normal levels. However, because the transplanted pancreas had been put just under the skin and it wasn't really connected to blood vessels correctly, it eventually degraded. And in a few days, the dog succumbed to diabetes again. But what was really remarkable about this, especially since the transplanted pancreas had been under the skin, this suggested that the pancreas might be releasing something. And maybe it was even a single molecule that could control blood sugar. The discovery set the stage for major breakthroughs that we'll explore later and in our next episodes. First, that transplantation could be a viable option for treating diabetes. And second, that a single molecule could be used as a treatment. And as we'll see, both approaches have revolutionized diabetes treatments, and their origins can be traced back to Minkowski's early experiments. So we know that the pancreas plays a major role in diabetes, but do you need the whole pancreas?
Exactly. And this brings us to the German scientist Paul Langerhans. In the 19th century, Germany had become one of the global leaders in making synthetic dyes for the textile industry. And it was driven by companies like BASF and Bayer. And even though the dyes were originally created to color cloth, they found an unexpected use in the medical industry, especially in histology or
Yeah.
the study of tissues under a microscope. So Langerhans had a superpower. What he had learned from his mentors was to make thin slices of organs and look at them under the microscope.
Right.
But you wouldn't be able to tell anything about the architecture
No.
of the organs unless you colored them, because you would just see clear and transparent
Yeah.
things. And so, what was remarkable about Langerhans' discovery was that by experimenting with dyes originally used for coloring cloth, he could now start to see very interesting patterns. And he found these cluster of cells that were distinct in the pancreas from the rest of the organ.
And they weren't the majority of the cells either, these little islands.
Right. And these cells later came to be known as the islets of Langerhans, which, as you will see, scientists found play a huge role in diabetes.
Right. Okay. So until now, the key milestones in our story are first, the pancreas is identified as the critical organ in diabetes. And next, the islets of Langerhans within the pancreas were recognized. And finally, scientists realized that the pancreas produces something that regulates blood sugar.
Yes, and this substance would later be called insulin. And this word derives from insula, the Latin word for islands, because this was realized to be made by these little islands that Langerhans had discovered. Pam, could you explain why insulin is so important and how it connects to blood sugar? I keep hearing people talk about blood sugar and about insulin, and I think it would be helpful to break it down just from the
Right.
basics.
Mhm.
What exactly is insulin and why does it play such an important role in controlling blood sugar? Let's start from first principles, Pam. Could you
Okay.
tell our audiences about it?
Right. So as we discussed, most of the food we eat gets broken down into sugar. That is the most important fuel for the 30 trillion cells in our body. And it turns out that insulin from the pancreas is the signal to all the cells to open sugar channels and pull in the sugar out of the bloodstream to use as fuel. So in the car analogy, insulin is the key to the fuel line so that the fuel can go through to the engine. And there are two reasons that it's dangerous if the sugar doesn't come out of the bloodstream. The first is the cells, the engines of our body can't function, right? That's the main thing. But also imagine a car owner who thinks, well, I don't seem to be getting much fuel into the engine, but I'll carry around lots of extra fuel, hoping or trying. I'll keep pouring some and see what happens. Well, of course, that doesn't help the engine if the fuel line is blocked.
It's also dangerous.
It's also really dangerous because the extra sugar in the bloodstream gets stuck places it doesn't belong, gets stuck on proteins, clogging blood vessels and tissues like the kidneys. And that's what leads to all the terrible complications of diabetes. So it was the realization that if we could identify the exact substance from the pancreatic islets, maybe it could be used to treat patients. What scientists in the early days would do is mash up the pancreas and try and inject the soup into an animal that had diabetes.
Would that work?
It really didn't work. And the reason is the islets are only 2% of the total pancreas. The other 98% is making enzymes that break down all our food. So if you mash it all together, you get the enzymes breaking down the insulin. I see an engineering challenge here. How do you get the stuff from the islet cells away without destroying it?
Of course, that was an interesting engineering challenge as you described it. And this is where we have to go to Canada. An orthopedist in Toronto, Frederick Banting, who's arguably one of the most interesting figures in our story. He's widely considered one of the most famous Canadians ever. Of course, perhaps even more than Justin Trudeau or Justin Bieber. Though it depends on whether you care more about science or popular culture. So now Banting had a childhood friend, Jane, who died of diabetes when she was just 14 years old. So that must have influenced him to start thinking about the disease. Banting decided to tackle the idea of isolating the islet cells, and he had learned a clever but difficult trick to try and accomplish this. He had read of a surgical technique where the main duct that goes from the pancreas into the gut can be tied or ligated, and this would cause all the other cells, the enzyme producing cells of the pancreas, to degenerate and die. And this would leave only the islet cells alive, the islets of Langerhans. But the timing was really crucial. You had to do this very carefully.
Right.
The insulin producing cells would just survive long enough to be useful. And Banting's mastery of this technique was what set the stage for one of the most important breakthroughs in medical history. Banting and his assistant Charles Best then turned to dogs for their experiment. The key experiment involves a dog called Marjorie. First, they of course replicated Minkowski's experiment. They removed the pancreas from Marjorie and she soon developed diabetes. And they knew that without doing anything, without any intervention, she would die.
Right.
So if they could extract and purify the islets or the extracts of the islets from another dog and inject it into Marjorie, she could survive. And so using this ligation method, Banting and Best purified the extract, created this magic potion, and this was able to keep Marjorie alive for up to 70 days, even though she no longer had a pancreas.
Yeah, that's pretty amazing. And so this was one of the first indications that a purified substance containing what would be called insulin could be used to treat dangerously high blood sugar. And this really gave birth to the monumental notion that this could eventually be a treatment for patients with diabetes.
Banting really wanted to quickly try this, of course, in humans. And by the time he was producing larger amounts of this substance from pigs, he was really excited and he was driven. But he needed someone who had more resources, and he was also struggling to gain recognition.
Yeah.
So he approached John MacLeod at the University of Toronto, who was a really much more well-established researcher with a lot of resources. So MacLeod didn't take him seriously at first. He saw him as an outsider and without formal research training, and he doubted.
And he was very disorganized.
Yes.
Yeah.
But even then, he just decided to give Banting a chance.
Yeah.
And what followed next was a famously tense collaboration. You know, as you said, MacLeod was meticulous, he was structured, methodical. Banting, on the other hand, was impatient and messy, and he was more driven by instinct.
Yeah.
I guess
Yeah.
that's true for many scientists.
Right.
So when he started working in MacLeod's lab, the place was full of dogs and experimental setups and urine samples everywhere, and far from the well-organized structure that MacLeod liked. And this drove MacLeod really crazy. He believed that Banting's approach lacked rigor and systematization, and their disagreements were legendary and even led to actually a physical fist fight.
Isn't that wild?
Meanwhile, word started to spread. Banting began to talk to others about his discoveries, hinting that he might have found a remarkable new treatment for diabetes. And this created a buzz in the scientific community, a mix of excitement and also skepticism. People were really beginning to wonder, could Banting really be on to something? And what happened next would reverberate across the world.
Ani, as we move forward with the story, I want to make clear that the diabetic patients we're pretty much talking about at this time were children with type 1 diabetes or juvenile diabetes. And these children uniformly died from the disease. Type 1 diabetes is an autoimmune disease, which means that our immune system, meant to protect us from pathogens, turns on itself. And
so it destroys the cells
that are making insulin.
Mm-hmm.
And until 100 years ago, with Banting and Best's work, basically no diabetic children lived to adulthood. So what you had were just desperate parents with dying children. And they so longed for any way to treat these kids.
And was there any way, what did they do before insulin? What was the treatment that was offered to these children?
It was a very imperfect treatment. They could extend lifespan by essentially limiting caloric intake, but really that was basically a form of starvation. So these kids were so tiny and malnourished. It was very sad.
And a 14-year-old boy who weighed only 65 pounds, Leonard Thompson, came to the Toronto General Hospital in the January of 1922. His story is one of the most remarkable moments in medical history.
So picture this. His mother is carrying him. He's a tiny, frail, skeletal child. It's absolutely heartbreaking to look at those pictures.
And this is when Banting and his team decided to try something really bold, because this has never been done before.
Yet,
They give Leonard the first injection of insulin. But here's the thing, it doesn't really work well. There's a tiny drop in sugar, but not enough to make a substantial difference. And so now they have to figure out what went wrong. But this is when James Collip, the biochemist in the team, he stepped in and he made a more pure, cleaner, and more effective extract.
Exactly.
Just a few weeks later, the second injection is given, and this time it works like magic. Leonard's condition improves dramatically, and his blood sugar stabilizes, his body starts recovering. In just a few months, his weight has doubled to 110 pounds, and people start calling it a miracle. And then, boom, word spreads like wildfire. Suddenly, everyone in North America, Europe, and beyond is talking about insulin. That was the moment insulin changed the world. Within a very short time, Banting and MacLeod received the Nobel Prize for Medicine, the fastest Nobel recognition in history at that time. But Banting wasn't happy. He believed that the Nobel really belonged to him and his assistant, Charles Best. And so he kind of showed his protest by giving half of his prize money to Charles Best. And MacLeod, on the other hand, donated half of his money to the biochemist James Collip.
I know. I really like that story of scientists sharing with their peers who they think deserve the recognition. And then one of the most moving accounts in medical history. Imagine a hospital ward that is filled with diabetic children in comas, literally at death's door. The doctors come in and start injecting the first child with insulin, and they move forward. And as they get to the sixth child, something amazing happens. The first child wakes up from their coma, and then the next, and then the next. And the parents are standing there watching their children who were almost dead open their eyes, sit up, and come back to life.
In our next episode, we will dive into how the discoveries, all the way from observations almost 3 millennia ago to the first insulin treatments, led to the current advances in treating diabetes, from human insulin and islet transplantation, and even the birth of the biotech industry.
Wouldn't it be cool if we could hear directly from the scientists who made that happen?
And that's exactly what we are going to do. So stay with us for the Discovery Dialogues podcast.